Assalamualaikum warahmatullahi wabarakatuh. Hello doctor, my name is Nurul Izzati binti Samharis. Together with Wong Junyang, we will be presenting on toxic food dyes. What is food dyes? Food dyes refers to food colorings or food additives that are added into food and beverages to impart and enhance their color. It is used to improve the visual appeal of processed food and products, and have become an ubiquitous part of the modern diet. These vibrant and enticing colorants are employed to make products more visually appealing. However, concerns have arisen regarding to their safety. The term toxic food dyes refers to certain synthetic color additives that have been scrutinized for their potential adverse health effects. In food and beverages industry, there are 7 common types of food dyes we use, which are brilliant blue, indigotine, fast green, erythrocin, allura red, tartrazine, and sunset yellow. In this presentation, Junyang and I will be discussing on acute and chronic toxicity, route of exposure, symptoms, metabolism, and mechanism of action from food dyes. For the route of exposure, most common route of exposure for food dyes is through oral ingestion. People consume food and beverages containing these additives, and the dyes are absorbed through the GI tract. However, dermal absorption occurs when handling food products containing food dyes, or when using personal care products that contain these colorants. There can be minimal exposure to food dyes through inhalation, especially in food production facilities where powdered dyes might be present in the air. For the mechanism of action, azo compounds are composed of an aromatic ring connected to a second naphthalene or benzene ring via an azo bond. Azo chemicals can enter the gut via the mouth or through the bile after parenteral administration. They are reduced by intestinal bacterial azoreductases and to a lesser degree by hepatic microsomal and cytosolic fraction enzymes. The reduction of the azo bond results in the formation of aromatic amines. This process is accompanied with a decrease in the dye's visible light absorbance and decolorization. When these aromatic amines produced come into contact with foods that contain nitrate or nitrate in the stomach, they can produce reactive oxygen species as part of their metabolism. The oxidative stress creates many adverse effects in humans after considerable dose of consumption. The next part is acute and chronic toxicity. Acute toxicity in food dyes is single exposure which only leads to allergic reactions, for example, tartrazine. And for chronic toxicity, is a continuous and prolonged exposure that can lead to carcinogenicity, for example, allura red. Nitrate or nitrate containing foods and aromatic amines can produce reactive oxygen species, including H2O2 and superoxide anion. Indigo carmine, an indirect ingestion of these amines with carcinogenic and genotoxic properties. Hayat et al. Examined the effects of sunset yellow and allura red, 2 azo dyes, on the structure and function of the kidneys and liver, the antioxidant status, and the genotoxic effect on the white blood cells of rats. When rats were given the distinct dyes, their serum levels of ALT and AST rise, suggesting obstructive injury to the liver tissue. Serum levels of creatinine, urea, and uric acid in the rats were significantly elevated. Malondialdehyde levels in rats dramatically rise, suggesting a rise in oxidative stress brought by an excess of ROS species. According to Junyang, tartrazine, sunset yellow, amaranth, and allura red can cause hatching difficulties and developmental abnormalities in zebrafish embryos, including cardiac edema, reduced heart rate, yolk sac edema, and spinal malformations. These developmental toxicity values in zebrafish embryos demonstrated that going beyond the allowed acceptable daily intake for azodice can have negative health effects, and pregnant women should be concerned about teratogenicity and embryotoxicity.
So good morning, everyone. My name is Junyang. So I'll be concentrating on the parts of symptoms and metabolism. So in terms of symptoms, for brilliant blue, it causes agitation and rash on the skin surface. It is listed by the US EPA as a human carcinogen because it can cause tumors and harm to DNAs in animals. Next, it displayed distinct changes in the distribution of peripheral blood leukocyte in rats, which were quite evident in the form of neutrophilia and lymphocytopenia according to the studies. For indigo carmine may result in brain tumors, skin rashes, nausea, and vomiting. It is also cancerous to animals and very harmful to DNA. And it produces a rare symptoms which is an idiosyncratic reaction such as rash, pruritus, and bronchoconstriction according to the monthly index of medical specialties. For erythrocin can cause hyperkinesis in children and possibly learning problems. Aside from that, it can increase the oxidative and nitrogenic stress, acetylcholinesterase activity, and release of
That's all from
pro-inflammatory
us. Thank you
cytokines in
for
the rat
listening
brain,
to our
causing biochemical
presentation.
changes,
Have a nice day.
worsen cognitive, and maybe neurobehavioral deterioration, according to the studies. For alurarate, a high dosage of alurarate can cause structural damage to the medial prefrontal cortex in rats. Symptoms-wise, it may cause loss of cells, dendritic trees, cortex volume, and maybe impairments in the learning and memory. It also enhances DDS-induced colitis and low-grade colonic inflammation in mice. For tartrazine typically can cause hypertech activity in children with or without attention deficit hyperactivity disorder. And also it can inhibit the activity of antioxidant enzymes in the colon and ileum. For sunset yellow, it can trigger allergic responses, particularly in those who are intolerant to aspirin. And also, according to a study in hyperactive children, a 50 milligram dose of sunset yellow, which is already above the ADI, it can result in an increase or acceleration of zinc excretion in the urine. Next, we are going to talk about the metabolism of all of the azodides. Generally, the microflora in the intestines involved in the reduction of food dye by azobond cleavage, and subsequently it will produce aromatic amines. And also, most importantly, it is only catalyzed by bacteria with azoreductase enzyme under anaerobic condition in the GI tract. So the released aromatic amines from the azo reduction may be absorbed by the intestine and subsequently excreted by the kidneys. Two metabolites may be released, which is sulfonylic acid and also aminopyrazolone. For brilliant blue, the GI tract will absorb only approximately 5% of the ingested brilliant blue, and the remaining portion will be excreted in the feces. For indigo carmine, the scientists produced studies on rats have shown that most of the indigo carmines and its metabolites, including the 5-sulfur-antranilic acid, which is a byproduct from the breakdown of indigo carmine, are eliminated in the feces with traces level detectable in the urine. For erythrocin, when it's administered as a single dosage to 14 male rats in an investigation, within 3 days, almost 55 to 72% of it was eliminated unchanged in the feces. For alurarate, azo reduction in the GI tract will produce P-crescidine sulfonic acid and is found in the feces and urine. For tartrazine is a little bit different because it involves the cytochrome P450 complex. So the CYP enzymes in the liver can reduce the tartrazine azodide, producing compounds such as aromatic amide, which may have the potential to cause cancer. For sunset yellow, the microorganism inside the intestine degrades the sunset yellow and the azo linkage in the intestinal gut to create sulfonylate acid, 1-amino 2-naphtol 6-sulfonic acid, and sulfonylate acid in its acetylated form. So, in conclusion, toxic food dyes encompasses a landscape where the quest for visually appealing and vibrant foods intersects with the concerns about the potential health risks. So, the safety assessment of food dyes involve rigorous testing is needed in order to determine their impact on human health. Therefore, consumers are encouraged to make informed choices by reading food labels, understanding the types of dyes used, and being mindful of the individual sensitivities.
Here are the list of the references Junyang and I used for the presentation. That's all from us. Thank you for listening, Doctor. Have a nice day.